All right, so we left off on this slide last time talking about the third way in which proteins interact with DNA. One is copying, and that was what most of chapter 14 is about. One is expressing, and that's what chapter 15 is about. And 3 is organizing, and that's what the very end of 14 is about, and does influence how DNA is replicated and how DNA is expressed. And I mentioned that we've got these protein octamers that are called histones that facilitate winding DNA up, condensing it down, shortening its length. And so this structure of DNA wrapped about 2.5 times around a histone is referred to as a nucleosome. And then you can see further condensing of these chromosomes down further and further with the use of other proteins. And so again, the way proteins function helping structure or organize DNA is by condensing DNA down. With just the DNA molecule, you have something that is incredibly long and difficult to deal with. And so condensing it down makes it easier to package within the cell. And then in order to access it, you just have to unwind it from its protein partner, from the histone. All right? Any questions about this? This is where we left off on Friday. It was like 3 days ago. And if your weekend was anything like my weekend, you have forgotten everything from Friday. Yeah. Cool. It's a Monday. It seems like it's a Monday. This is my second class of the day so far, and I guess it's week 5, but it's only going to get worse. It's only going to get worse. All right. So how does prokaryotic packaging differ from that of eukaryotes? All right. First point. Prokaryotic cells have much fewer proteins associated with the DNA than eukaryotes do, which makes sense. Eukaryotic cells have way more genetic material, and so you have to worry about organization far more than with something with far less information, right? It's like if you have, one, birthday card, like you don't really need to worry about how you display those. You just put it there and you feel a little sad that only one person gave you a birthday card. But if you have 50 birthday cards, now you're like, how do I organize these in such a way that displays them all and shows their importance, right? Any of you do that with birthday cards? I used to, but people don't send me birthday cards anymore. So, yeah, sad. All right. Another difference, bacterial chromosomes tend to be circular. Not all bacterial chromosomes are circular. There are some that are linear. However, most are circular, and they're suspended in the cytoplasm. They are not packaged in a nucleus. So again, you don't have the same constraints that you do in eukaryotic cells. Eukaryotic cells, you're dealing with a lot more information, and you've got to package it in a structure that only takes up a small percentage of the cell's volume. In a bacterial cell, you've got way less information, and you don't need to package it in a small percentage of the overall volume. Okay, it takes up most of the volume of the cell. In prokaryotic cells, replication happens at a single origin. You have one origin of replication, and then it'll continue until the forks converge, until the forks meet at a single site. Whereas in eukaryotic replication, you have multiple origins of replication. Another unique and important difference is many bacterial cells have accessory chromosomes. In addition to having their main circular chromosome, they have additional DNA molecules called plasmids that occupy machinery necessary for copying the main chromosome. And so the bacteria will replicate these plasmids when it's dividing, just as it does its circular chromosome. And so you just have a different feel to how the DNA is packaged, how the DNA is copied, and how the DNA is expressed, which we'll talk about this week, between prokaryotic and eukaryotic cells. All right? So, I don't want you to equate less information with simple, okay? There is nothing simple about prokaryotic cells. They are incredibly complex and sophisticated. They are just less complex and less sophisticated than eukaryotic cells, right? They are still incredibly complex. No questions? Have I said good morning? Good morning. Hope you all are enjoying yourself so far. So here is the replication in a bacterial cell. You have a single origin of replication, and the forks move as helicase unzips the molecules from each other all the way into a point in which your two replication forks converge, at which point your replication is complete, and you have two chromosomes with one old and one new strand. What do we call this model of replication? Semi-conservative. And at each replication fork, you have a leading strand and a lagging strand. And what do we call that model of DNA replication? Semi-discontinuous. Semi-discontinuous. One leading strand and one lagging strand. All right? Yeah, Joey.
So, am I right in understanding that, like, a DNA molecule by itself is very dependent on all the proteins around it that make it useful, right?
Yes.
Okay.
Yeah, it's the same thing, like information is only valuable if you have a way to read it and use it.
Okay.
Right?
So, like, when scientists try to describe how, like, the first DNA molecules came about, I feel like is there, like, a lack of explanation for, like, even if you could get a DNA molecule, what good is it if you don't have all the machinery?
Yes, you have some issues. Yeah, and we'll talk about different views on the original machinery of life. I think very few people would argue that DNA is your original information storage molecule. That DNA was something that developed later on because of its stability.
Okay.
Yeah, and it was not the original machinery. Yeah, we'll have some good discussions there. All right, any other questions before we transition to our quiz? Can we not take it? You absolutely can choose not to take the quiz. Yep, you're right. Yeah, that is well within your rights. All right, so let's get this paused. All right, so as we work through chapter 15, two people in the class let me know that there's something wrong with the slides on Canvas, that they're not coming over properly or not working for some people. It worked for you?
Well, you just have to X out of the master slide.
Oh, okay. Oh, I got you. Oh, man. Okay, so yeah, if you download the file, that's weird that it left it in the format it was in when I saved the file. So it's over in View, Master Slide. So if you just click on the tab that says View and go over to Normal, it'll give you the normal slides. Okay. All right. So the first set of questions that we go over for this chapter, I want to do something a little bit different. And I actually want to use one of these as our lecture break questions. And I know we just took a quiz, and it seems weird to go from a quiz immediately into a lecture break. But I hope that'll actually facilitate our discussion. So the first four questions: what are the 3 steps of transcription? Next, what are the 3 steps of transcript modification? Third, what is unique about transcription in eukaryotes? And then lastly, how does transcript modification add complexity? Okay, so these are the first 4 questions we are going to interact with. And what I would like for you to do now is getting into your groups, which y'all should be in. After Friday's exam, y'all can sit wherever you want. You got to maintain groups of 5 or 6 people, but you can go back to sitting next to, your BFF or whatever you call people these days. I'm old. So, and what I'd like for you to do is discuss this 4th question. How does transcript modification add complexity? Okay, take a couple of minutes, talk about this with those in your group, and then we're going to talk about this collectively, and we'll go in and fill in some of the gaps and hit some of these, what's going on during transcription, what's going on during modification, how is this unique in eukaryotes, and then address this question actually as a whole. All right? So take 2 minutes, talk through this. How does transcript modification add complexity? 2 minutes, starting now.
By transcript modification, you mean
The processing.
The fact
So,
that
so splicing, polyadenylation, capping.
It's not like when they remove the introns.
Yes, yes. Yep, absolutely.
Okay. Well,
Yes,
I think
yes.
for starters, it's like, I mean, I always just pictured like, okay, like
I have children
DNA molecule
that are always grumbling about it. You gotta hear exactly what they say so you can use it against them.
the exact sequence of the acids, right? But then,
You know, my
like,
son told my
this
wife
added
2 days ago that
it's
he
like,
wanted
the
to
DNA
do.
molecule is more like
He's messed up.
processed code.
That is messed up.
why did he say that?
Cause I mean, he was being mean.
Well, like what's the filter?
I don't know. Yeah, I don't know. I wasn't there.
So it can affect how the same DNA can be expressed.
You got about 30 seconds to finish your thoughts. Get somebody to represent your group to the class as a whole.
Okay. And then I think just in general, it also just is another step in the process. Because now you're not just worried about copying the DNA. You have to copy the DNA as the process. It's just another step in the process. So it not just adds complexity to how the DNA is expressed, but it adds complexity to how the DNA is.
All right. So because there isn't a lot of variation in how you can address this question correctly, we're just going to take groups to volunteer to answer this question rather than going group by group. So I see a hand from group 6. What do you got for us, Joey?
It's like something that Alyssa shared, which is really cool. So like all cells in your body have the same genetic code.
They do.
So they're all the same DNA, but obviously that's being expressed differently.
Yes.
Your cells look differently. And so this process, like we're all taking the same genetic code, but the way that that genetic code is getting rewritten or reorganized is going to change how those proteins are formed.
Yeah, you have to keep in mind that there are basically 3 processes to expressing that information, right? You've got to transcribe it, at least for eukaryotic cells. You have to, well, you transcribe it for prokaryotic cells as well. You've got to transcribe it to take it from DNA into RNA. And then you've got to process that piece of RNA. And then you've got to translate that RNA into protein. So you have 3 processes in order to express the information in the genome. And any way in which you can make it to where you have more possible products from the same piece of information, that's going to add complexity, right? So from a single gene, because of specifically splicing, being able to, as you cut out introns, being able to change which parts of that immature mRNA get cut out, and that varies with alternative splicing, you can, from a single gene, produce several different protein products, therefore add to some complexity. And so I think currently the numbers are, there are about 28,000 genes in the human genome, and we have 4 billion bases. And so actually very little, less than 1% of our genome is actively being expressed as protein. And the rest of the genome is functioning in other ways. We'll talk about that. Yeah, Cindy. A lot of them are being transcribed into different types of RNA. Like not mRNA or ribosomal RNA or transfer RNA, but are being transcribed into other types of RNA that function either in the nucleus or in the cytoplasm for various regulatory roles. A lot of them controlling rates of expression of proteins. Yeah, that's a good question. Yeah, Charles.
I kind of wonder that yes, but is it? Well, no, it's really. I hear a lot, especially from creationists, that you can't add DNA. Like organisms cannot gain.
You can't add information.
Add information. Is that
Okay.
true? And why is it true? And why can't you?
I don't know. So it probably would be better to say that we don't have evidence of adding information and it working. Is it mechanically possible? Yes. I mean, there are mechanisms by which you can increase the length of a genome. But we don't have good evidence that it can be used to fuel diversity, to fuel, making different forms. Yeah. And then, and we'll get to that. That's a question that we'll deal with quite a bit. Any other questions or, comments of how this adds complexity? Okay. In a lot of ways, transcription is very similar to what's going on when you are replicating the DNA. The difference is, rather than reading a template strand and writing a new DNA, what's the term I'm looking for? Sequence, but I'm reading the template and writing the congruent or the complementary, a complement. Rather than writing a DNA complement, you're writing an RNA complement. So this process of transcription is you're writing an RNA complement to your DNA template. And so this process involves 3 steps. The first is initiation. And during initiation, several things happen. We've got transcription factors, abbreviated TFs, bind to the promoter region. I heard some of you before class talking about the Tata box, right? And getting some joy out of that name. Tata box, I've heard it pronounced both ways. I don't know the way it was pronounced by the person who initially discovered it. And it's basically just a sequence that is TATAA. And it gives you a nice Tata box as part of the promoter. And that helps RNA polymerase to know where to bind. DNA unwinds, similar to when DNA is copied. You still need to pull the 2 strands apart, right? It's still something that needs to happen. Now, you don't need helicase to do it because RNA polymerase actually does it itself. RNA polymerase, unlike DNA polymerase, is self-priming. And this is where primase that we saw laying that RNA primer, it's a type of RNA polymerase. Okay? And so RNA polymerase is self-priming. It's physically attached to the template strand, just like DNA polymerase is physically attached to the template strand when DNA is being copied, and it reads the template and writes a complement. In this case, it writes an entirely RNA complement to that template strand. Yeah, I mean, obviously this is part of gene expression, so knowing that you transcribe DNA into RNA is important, but specifically that it's RNA, it's an incredibly important piece of information. Don't forget that. And it's unfortunate that this isn't going to appear on a midterm for, 6 weeks from now, but don't forget it over these next 6 weeks. It's an important piece. The next step is elongation. So RNA polymerase attaches during initiation and starts to transcribe. During elongation, RNA polymerase 2, this is in the case of writing mRNA, moves along the template strand, writing an RNA complement, writing all the way until it gets to a termination sequence. Okay, so elongation can be really, really long if your transcript is really long, or it can be pretty short if your transcript is rather short. Most of the transcripts are going to be really long and then get modified in some way. And what's the third step? So you have initiation, elongation, termination. And so during termination, the RNA transcript and the RNA polymerase are released. They fall off the DNA template. And now your RNA is free. It's free to be modified, which is the second part of DNA expression, right? Of gene expression. So it's free to be modified. It's free to be matured, if you will. Another way of modification or maturation. 2 terms for the same process. Any questions here? Yeah, Joey.
Is this like for the, are what we're seeing right now good for like eukaryotes and prokaryotes, or just
Yes,
just
yes.
Okay,
Uh-huh.
so we're keeping it to the.
Now, the promoter's a little bit different in eukaryotic cells than it is in prokaryotic cells. You don't find the Tata box in eukaryotic cells, but you find promoters similar to
Okay,
that.
okay.
And then these processes are the same. Yeah,
Okay.
yeah, the
What's the
same.
difference between a Tata box and like the AUG or the beginning?
Oh yeah, AUG is a codon. So the Tata box is also a sequence of bases, but it's not specifically 3 bases like the codons are, and it's not starting a process. It's really just functioning to get RNA polymerase to bind to the correct spot. RNA polymerase starts the process. The Tata box is kind of a placement, whereas AUG, that start codon, that becomes the first part of the protein. The Tata box does not become the first part of the transcript. The Tata box just helps RNA polymerase bind downstream. It does not become part of the transcript itself.
So all proteins have the first thing in it?
Methionine. Yep, methionine is the first amino acid of every polypeptide that our cell makes. Now, many of those get modified and that methionine gets cut off, but initially methionine is the first amino acid of every polypeptide. Yeah. And so here's this process of transcription. And so here you have this box, and it says of a eukaryotic protein, the box is a little bit different. And so here you've got this, again, this promoter where transcription factors bind and help to facilitate RNA polymerase binds to the correct place and starts transcribing. And once RNA polymerase gets to this red region, which is your gene, elongation is happening. So initiation is going on while your RNA polymerase may bind part of the promoter, the very end of the promoter, not the Tata box, but the very end of the promoter. Once it gets to the end there and gets to this red, the part that's actually the gene, the active part of the transcription element, now elongation happens until you get to a termination factor. And so here's RNA polymerase just going through doing what RNA polymerase does best, which happens to also be the thing that DNA polymerase does best. The difference being that RNA polymerase is self-priming, which means what? It doesn't need a primer, so it can add the first nucleotide. It can't, unlike DNA polymerase, it can add a nucleotide even where there is not a free carbon 3. It can add that first nucleotide. All right. So what are the 3 steps of transcript modification? Or again, you could call this transcript maturation. So at the end of transcription, you do not have mRNA. You have what we call pre-mRNA. It is not matured. It's not been modified. It is not ready to be translated yet. And so it has to mature. It has to be developed into mRNA. And there are 3 parts of this process. First off is a guanine nucleotide is added backward at the 5' end. So at the very beginning of your pre-mRNA, you add a guanine nucleotide on backwards. And this provides this site that ribosomes recognize and attach to do the 3rd part of gene expression, which is translation, which we probably won't talk about today and we'll get to on Wednesday. Another part of this modification is there are proteins that remove the 3' end, the very end of your pre-mRNA, and replace it with a whole bunch of adenine nucleotides, anywhere from 50 to 250 adenine nucleotides. And this functions to mark this as a mature RNA and also to provide a place that's non-functional for enzymes that chew up nucleic acids to chew on before it gets to something really important. It's like having a tail as a lizard that can fall off, right, and help you get away. Which I think provided the idea behind the name of it, the poly-A tail. Plus it's at the end. And then any regions within, in between the 5' end and the 3' end, so in between the beginning and the end, any sections in there that do not need to be translated have to be removed. And you call those introns. sections of the pre-mRNA that are not translated. Okay, you call those introns, and you cut those out. These are cut out by small nucleoprotein particles that are called spliceosomes. Yeah.
Agreed. So the guanine nucleotide is added on the one end, and then are those 50 to 250
Added on the other end.
Other end.
Yep,
Okay.
at the 3' end. Yep, the very opposite end. And then in the middle, the processing is cutting out introns, cutting out sections that aren't going to be translated for that particular protein product. But again, because of alternative splicing, you can cut out different sections to produce different proteins. Yeah, Alyssa.
So 2 questions. Why were the introns there in the first place if they're not going to be transcribed?
Because, well, they are transcribed, they're not translated.
Well, that's what I meant. Yeah.
Yeah, because something that's an intron for one protein may be an exon for another.
Okay. And how does the specific cell know what is an intron and what's not?
It's an excellent question. There are particular sequences that show you where you transition from an exon to an intron, and then which ones get cut out are processed by different RNAs. And that's what Sydney was asking, what does the rest of the genome actually do if it's not being actively made into protein? A lot of it's being transcribed into RNA, and some of those are controlling this process to making sure the right portions get cut out for the particular protein product you need.
Okay.
So like if the cell needs, say, protein A, it'll cut out introns that aren't part of the protein A, that don't get translated to become part of protein A. But if it needs protein B, it'll have different exons, introns, and so it'll cut out different portions of that pre-mRNA. And a lot of that is controlled by RNAs, controlling which parts get cut out for whatever the needed protein product is.
Okay. And that's affected by like the cell itself, like intrinsically, or is that something that's exosomal?
It could be either. It could be something controlled entirely by the cell itself, or something that was triggered by an external signal.
Okay.
Yeah, it varies. Yeah, some of it's just homeostatic, mechanisms. The cell has to, maintain certain internal features, and so it does it itself. But a lot of it is external control. Cell gets signaled in a certain way to start producing something, and it'll start producing that. And so here's the processing. Here's our 5' cap. Again, guanine attached backwards. And so you can see guanine followed by its 3 phosphate groups and then the next nucleotide, rather t'n being a phosphate group, then the nucleotide. So this is attached on backwards. And then at the 3' end, you cut off the 3' end and add a poly A tail, a string of 50 to 250 adenine bases at the very end. And again, this is to protect the RNA from nucleases, enzymes that eat nucleic acids. And then here's the last part of the processing, splicing. And it's not necessarily in that order, like first you cap, then you polyadenylate, then you splice. But it's just those are the 3 parts. And so here are small nuclear riboprotein particles that are made up of RNAs and proteins that grab on and cut at very specific places. Cut out the introns, leave the exons that are going to be translated. So all of this is happening inside of the nucleus. Transcription happens inside of the nucleus. RNA processing happens inside of the nucleus, at least in eukaryotic cells. Why wouldn't it happen inside the nucleus in a prokaryotic cell?
They don't have one.
Because they don't have one. Look at that. Critical thinking at its finest. And so here, the intron has been completely spliced out, and then these bases can be recycled and used in, further transcription. Or they can be recycled and used for energy, for harnessing energy. All right, now your RNA is mature. It's now mRNA, and it can be used for what? Translation, the last part. But again, we won't get to that today. But that's foreshadowing of what's to come. All right, so what is unique about transcription in eukaryotes? Man, this just keeps coming back. So I was wrong when I told you that the Tata box is a prokaryotic promoter. It is a eukaryotic promoter, and the prokaryotic promoter is a little bit different than being TATAAA. So the Tata box, the promoter sequence is different in eukaryotic cells than it is in prokaryotic cells. In eukaryotic DNA, RNA polymerase II needs transcription factors to bind to the correct place. The eukaryotic genome is more complex and more sophisticated, and so you need higher levels of control over what gets transcribed because the genome's a lot bigger. You can't transcribe it all. It'd be too energetically costly, at least not at any given time. Eukaryotic DNA lacks terminator sequences. So in prokaryotic genomes, you get to a particular place in transcription, and you get to a terminator sequence that gets it all to fall apart. In eukaryotic DNA, it has to be controlled by other mechanisms telling the RNA polymerase where to stop. And then the last one, the promoters for non-coding RNA. What we mean by non-coding is RNAs that are not mRNA. Okay, so any non-coding RNA is any RNA that is not mRNA would be considered a non-coding RNA. So the promoters for non-coding RNA genes are specific to the RNA polymerase type. So basically what this is saying is there is a different RNA polymerase for each type of RNA. Every type of RNA has a different RNA polymerase. And so RNA polymerase 3 writes all of your tRNAs and one of the 4 ribosomal RNAs. And then RNA polymerase 1 writes or transcribes the other 3 ribosomal RNAs. And then what does RNA polymerase 2 transcribe? Genes, the coding RNA, the mRNA. Or in eukaryotes, the pre-mRNA that then gets processed to become mRNA. That's important. And I know you're like, man, you told me this chapter is not on this midterm, right? So you're like, I'm partially checked out. I'll get to it when I can get to it, which I think is going to be in week 10, which is like 2 or 3 weeks after spring break. But don't forget this. This is really, really essential to know that each type of RNA has a different RNA polymerase. Okay. Any questions about this? Looks like we're going to have plenty of time to get to our 4th and final of this first set of questions, and then we'll be in a good position on Wednesday to get through most of the remainder of chapter 15. Won't get through it all. That's okay. We use our Friday a little bit differently. All right. And so here's just the overall process of gene expression. Here's transcription, where you take your template strand and you write an RNA complement. And if this is then going to go and get translated, what type of RNA is this? mRNA, coding RNA. And then it gets translated into protein. And see, it's referencing something that we'll talk more about on Wednesday, and these codons. These words that are composed of 3 letters. Okay? Words that are composed of 3 letters that then get translated into a word that consists of a single letter in a protein language. Okay? So in the nucleic acid language, your words consist of 3 letters. In your protein language, your words consist of a single letter, a single amino acid, making up the primary sequence. All right. So the last question that we will deal with today. How does transcription modification add complexity? And group 6 talked about this having to do primarily with the alternative splicing, where you can take a single gene and you can generate more than a single protein product. Okay, you can take a single coding RNA and generate multiple proteins. And so here's kind of walking this through step by step, dealing with this. So a single pre-mRNA can be spliced in a number of different ways. And remember, it's not a mature mRNA until it's done all 3 parts of processing: capping, polyadenylation, and splicing. So it's got to have those introns cut out as part of that maturation process. Yeah, listen.
So prokaryotic cells don't do this. They just go straight from transcription
To translation. And since there's no nucleus, both happen at the same time. There's no nucleus, so there's no separation of transcription and translation. Transcription and translation happen at the exact same time in prokaryotic cells. As something's being transcribed, it's being translated.
That's stressful.
It is stressful, and it gives you significantly less control over how much protein product you make. Eukaryotic cells have much more control over how much protein product they make, which is good. We'll talk about this on Wednesday, why that's important. So yeah, this increases the size of what we call the proteome, which is a collection of all proteins that an organism makes without increasing the size of the genome, which is a collection of all of the genes that an organism has. All right. Now, in addition to having splicing, where you cut out introns, you can actually rearrange your exons as well. And so you may have a sequence a squiggle is not an indication of the actual structure of the transcript, rather an indication of where we have exons and where we have introns. So this would be exon 1, this would be exon 2, this would be exon 3, and this would be exon 4. Okay? And so these squiggles are introns that are going to get cut out. So in addition to splicing these, in addition to cutting these out, you could also generate a mRNA that has E1, then E2, then E3, then E4. Or you could generate something that had E1, then E3, then E2, and then E4. Okay, and so there are ways in which you can scramble these exons as well, and we call this exon shuffling. Shuffling the exons, yeah.
If that's the case, then what's the point of the genome in the first place if it's going to be just switched around?
Well, it's not always switched around. It has the potential to be written in the same order or to be written in a different order. So again, the whole point of that is increasing your number of protein products without having a genome that's even longer. So our entire genome consists of 4 billion bases. Mm-hmm. But less than 1% of that actually represents sections that become protein product. Mm. But if you couldn't splice pre-mRNAs in different ways, and you couldn't, mess around with other things, that portion would need to take up a much higher percentage. Which, since we know that the rest of the genome still functions, it would mean that the entire genome would need to be bigger. And would make it even more of a challenge to fit that amount of information in the nucleus of every cell. So you already have 6 feet of information in the nucleus of every cell in your body. And so if you've got to, let's just say conservatively that the average gene can be spliced in 5 different ways. And I think that would be a conservative estimation. If it couldn't be spliced in alternative ways, then you would need 5 times as many genes, right? Which would make your overall genome significantly larger than it already is.
And is that controlled by the RNAs?
Which parts get cut out and when?
Or like the arrangement of it?
Yes. Yep. Yep. And so here's just an illustration of this alternative splicing. Here you have a fairly complex pre-mRNA with 12 different exons. And notice that none of your introns so if you express all 12 exons, the portions in between those 12 exons never become part of a mature mRNA. However, there is a possibility to only take, say, in this case we're taking 1 and then 3 through 11. So we're skipping 2 and 12. And so now you actually have this portion becoming an intron for that product, this whole portion here becoming an intron for this product. And so you can take some things and cut them in different ways to produce different products. The question I think you're dealing with is why even have these regions in the first place, right? That never become exons, right? Because you have this section here, and 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 so on and so forth, that never function as exons. Why are they there if their entire focus is to only function as introns? And they're probably place setters to ensure that the cut happens correctly and doesn't actually cut out any portion of the exons. Whereas if you had your exons being completely adjacent to each other without any intermediate space, and you then have to cut this out, you may end up cutting out a portion of exon 1 to cut out exon 2. And so these functions is like, place setters, protections, to ensure that you can cut this out correctly without actually taking out any portion of exon 1. Right? Make sense? And so to kind of sum up this question that I had you all discuss as a group, how does RNA processing lead to added complexity? It just basically, it makes it to where your proteome, your total amount of all of your protein products, can be as big as it is without having to have a larger genome, without having to have more DNA inside of every nucleus. All right? Okay. We'll pick up here on Wednesday. We'll talk about junk DNA. We'll talk about translation.